My name is Kaleb and I'm going to be presenting today with Parker Fitzgerald, and we are going to be presenting on the biological and phytochemical analysis of Podophyllum peltatum fruit. So a little background on the mayapple fruit. As I said, scientifically it's known as Podophyllum peltatum. More commonly it's known as mayapple or mandrake. This is a plant that's native to the northeastern United States, including Michigan. And that's actually one of the reasons why we're studying it. We work in an ethnobotanical research lab under the guidance of Dr. Doyle, and the research that goes on inside of our lab focuses mainly on the different fruits native to the surrounding area, including all of Michigan. And so this is a fruit that we took a special interest in with this project. Currently this fruit does have some ethnobotanical uses, specifically podophyllum is derived from the plant itself. It's found mostly in the roots and rhizomes of the plant. It's also found in the leaves, the stem itself, as well as the unripened fruit. And this is a molecule that is used as a topical to help treat genital warts caused by the human papillomavirus. It's also used as a derivative for two anti-cancer drugs or chemotherapeutic drugs known as tinipicide and etoposide. But we're actually not looking at the podophyllum itself. So as I mentioned, the unripened fruit is contaminated with the podophyllum. However, when the fruit ripens, it becomes a bright yellow color and no longer contains this podophyllum. And that's one of the main reasons Parker and I decided to investigate this fruit further. So I focused more on the collection and extraction process, and then Parker kind of focused more on the finer details of finding specific components within the fruit itself. So I'll kind of go through that and then he'll go through his piece. So for the extraction process, as I said, I would go out into the field. I would collect the fruit. It grows in these plants and are kind of low to the ground, and they are easily and readily consumed by ground animals such as squirrels, chipmunks, other animals, raccoons like that. So they're kind of hard to get the ripened fruit itself. However, when we do collect the ripened fruits, we collect them, we bring them into the lab, we dry them out inside of a dryer at 37 degrees Celsius, and then we put them through something known as a hammer mill, essentially just to ground them into a fine powder. This powdered fruit is then placed inside of methanol for a process known as maceration, and this allows us to essentially extract all the components out of the fruit powder itself. This methanolic maceration extract is then ran through a filter and then it's lyophilized and evaporated so that we can collect the components themselves from the methanol. We are currently working with two different extracts, and essentially what I mean with that is we just have two separate fruit collections from two separate summers. So they're the same fruits, they were just collected at two different periods in time. From these crude extracts that we collected and then isolated and dried as I just described, we tested them for biological activity in 12 different assays in partnership with a lab called Amway. These 12 different things, or assays as I should say, were different therapeutic categories, and we tested them for things such as anti-inflammatory properties and anti-aging properties, which the mayapple fruit does show positive biological activity in these two, specifically in the NF-κB pathway as well as the S6 kinase pathway. Additionally, we ran an on-campus cell assay to test for glucose uptake, and we discovered that the crude extract of the fruit does in fact inhibit glucose uptake and therefore does serve as some type of an anti-cancer drug in that regard. Moving on from that, we then focused more on the phytochemistry and trying to isolate particular compounds from the fruit itself. So for my portion of it, I essentially used thin layer chromatography to try to develop an ideal elution method. TLC essentially focuses on the different polarities of compounds within a crude mixture and using these compounds in order to separate out the components within that crude mix. Using the mayapple fruit, I discovered that the ideal elution mixture for this fruit is 100% dichloromethane for normal phase TLC and 1:1 water to acetonitrile for reverse phase TLC. I then use iodine and shortwave UV radiation in order to visualize the components within the crude mixture, the separated crude mixture. And I use these as these are non-destructive methods and allow us to go back and look at different plates over time. Following this initial phase of looking at crude extracts, we then wanted to partition the extracts further based upon their polarity. So this involves liquid-liquid partitioning, which essentially we take the crude extract, we separate it into a very hydrophilic component and a very lipophilic component, and then we take the hydrophilic component and separate that further into a very polar component and a semi-nonpolar component. So as you can see on this diagram here, we take the crude extract, separate it into an aqueous and a petroleum ether extract, and then take the aqueous extract and partition that further into an aqueous and an ethyl acetate partition in order to get those 3 levels of polarity. These are some of the TLC plates that I was able to develop from the ethyl acetate partition. Specifically, we went forward with the ethyl acetate partition because, as you can see from these plates, these showed pretty solid separation and allowed for us to be able to try to isolate specific compounds from this for further investigation.
Okay, hi, I'm Parker Fitzgerald, and like Caleb said, for my portion of the project we did here, I analyzed the ethyl acetate samples using HPLC, or high pressure liquid chromatography. So what I initially did was I dissolved the ethyl acetate solids in methanol, creating samples of about 1 milligram per milliliter in concentration. What I would do with these samples next was I would inject them into the HPLC, and what this essentially does is it injects them onto a column that has solvent flowing through it. The column I used was nonpolar, so what that means is that nonpolar compounds in the sample will elute off the column more slowly than a more polar compound would. After the compounds elute off the column, they are detected using the detector you can kind of see here in the diagram on the left. And that detection will measure the absorbance of these compounds at different wavelengths of light. And the spectra there, each peak corresponds to a compound in the sample. Going back to kind of what Caleb said, that podophyllum is associated with the mayapple, that was investigated first. So here I have 3 spectra that were run: the crude extract for the mayapple, podophyllum by itself, and a combined sample with the crude extract and podophyllum. What these spectra show is that podophyllum is likely not in the crude extract because this peak here for podophyllum is not present in the crude extract spectra. And also, some supporting evidence for this conclusion is that if podophyllum were present in the crude extract, this spectra would resemble the one on the far right a little more similarly. After the analysis of the podophyllum, I then took the ethyl acetate samples that Caleb had obtained and analyzed them using HPLC. To determine what samples that I would run on HPLC, I looked back at the TLC plates that Caleb had performed. What I was looking for on those TLC plates was good spots with decent separation from the others, indicating that the compounds were separated from each other. I have here the 4 spectra on the right. Those are just some of the spectra for some of the samples that looked promising from the TLC plates. And here I have the 109 crude extract from the mayapple, ethyl acetate fraction, and a methanol blank here to compare to the spectra that I ran. After deciding what samples looked promising to run on HPLC, I then worked to develop a method of HPLC that would maximize the peak separation in the spectra. So what I did with this was I toyed around with the variables of flow rate, solvent gradient, and runtime to maximize that peak separation. What I ended up deciding with the method was that a runtime of 30 minutes, a flow rate of 0.75 milliliters per minute, an injection volume of 1 microliter, and a gradient for the solvent that transitioned from concentration of 50% methanol and 50% water to a concentration of 100% methanol over about 13 minutes. This seemed to work the best, and I ended up working with the ethyl acetate 21 sample because I had a large amount of sample to work with, and I wanted to develop a sound method with this sample before I moved on to some of the better ethyl acetate samples. So moving on, I ran the EA21 sample on the HPLC, but instead of just having the sample elute off the column and go into a waste container, I actually used that tube that goes to the waste container and directed it towards this micro well plate. And then I only used the micro well plate one time because I wasn't able to collect a large volume of samples, so then I upped the scale and used 10 milliliter test tubes following that. Here I just have shown some of these are from some of the wells in the micro well plate to see if I was able to isolate compounds from that elution fraction. Here, this is the one that was the most promising because you can see this really tall singular peak indicating that there's only one compound in that collection. So going on further, I then ran the ethyl acetate 21 sample a total of 7 times with collection of the elution, and that seemed to work pretty well. And I analyzed the eluate here. You can see in the 2 spectra on the right, those are from the 2 elution fractions seen here. And as you can see, there's a singular peak in them both. The EA21 T15 to T17 has a small second peak, but the one above has a good singular peak indicating that the isolation of the compound was fairly successful. So that is what I've done up to this point, and now I'm at the point where I would like to analyze these elution fractions using nuclear magnetic resonance spectroscopy. And what this analysis technique will do is it will help to indicate some of the structure and or identity of the compounds that I've isolated. And so for the next steps, NMR is the big one for me to see if I can figure out anything about the structure and identity of these compounds. And it was also discussed that using the HPLC to isolate more compounds from some of the ethyl acetate samples would be useful. And there's also preparative TLC, which would again help further this project. And also collection of additional fruit to provide more extract for future experimentation would also be very helpful. Here are acknowledgements and sources.